Okay, so for this question, I wanted to look at the 2019 paper, and it's the AS paper, which is part of the content which you will actually need to know for the AS part of the course, but also the A-level part of the course. So it's always good to practice some AS questions. So what I'm going to do in this session is I'm going to look at this question, which is linked to the topic area that I have put onto my website to help you when you are revising the content. So here you can see my website, which is ukbiotutor.com, and on there is lots of information about the use of microscopes. So it's the cell structure, it's got information about the microscopes that you need to know. It's also got information about how to do the different types of preparations of your slides, as well as how to use an eyepiece graticule, and also information about what you're looking at with the different dyes, and when you are using the magnification calculation. So all of that is there on my website, ukbiotutor.com, for you to use. And I'm going to show you how to answer this type of question, which is a question that comes up quite a lot, but it's important that you have the skills to be able to do that. So the first thing we have to note when looking at this, it says, right, okay, on the insert. Now, what we have is a mammalian blood smear. So let's have a look at the insert then. So here we have the insert, and in that insert, you can quite clearly see there are some blood cells. Now, they've given us A, B, C, and D. So they've labeled different cells. So they've already distinguished different cells. Now, one of the things that I love about this is I love to annotate around diagrams that I am looking at because it helps me to refresh what I'm talking about. So what can I see? Well, straight away, I know that this is a blood smear because they told me. And therefore, I need to think about the different cells that I have. And I know I've got erythrocytes. And I'm going to label this one because I know that this is an erythrocyte. An erythrocyte is the correct terminology for those of you who are just starting this from GCSE. It's the name we give for the red blood cell. So you need to stop saying red blood cell. You now need to be saying it's an erythrocyte. We know the erythrocyte is obviously not shaped like that. The erythrocyte, let's try that again, is biconcave in shape. So it can look like that if the slide has actually been done in a way that the smear shows you it on its side, or you may see it looking a bit like a donut, which is what we can kind of see in this diagram. There's the outside of it, and it's a little bit lighter inside. So what we've got is the donut shape of the erythrocyte. So I know that B is an erythrocyte and there's lots of them. Now I have some other cells. I've got these big cells, the A, the B, and the C. Now they all look slightly different. So I need to think about, well, what am I looking at here? I'm looking at cells. I can see that they're cells because they have different stains to them. And that therefore allows me to identify that these are probably my white blood cells. But let's just go back and see what the question actually wants us to do. So figure 2 on the insert is a photomicrograph of a mammalian blood smear. Calculate the image length. So I want to know the image length, and again, highlighting in micrometers represented by the scale bar. So in a diagram, you should be given either, I'll highlight these here, the magnification, which they have given us in the picture here, which is times 1250, or they will give you a scale bar, as you can see here. So this is my scale bar they want me to use in this question. So to use this scale bar, I would need to use a ruler to measure that. Now, obviously, as I am doing this, I'm doing this on my screen, and so therefore, what I am doing, one second. What I'm doing with this one, because I'm using my screen, is I'm not going to have it exactly as it was in the exam. So you have to be aware, if you have downloaded this paper and you are using it to try and practice, you might not get exactly what the mark scheme wants you to get. So you need to use the ruler that you have in front of you, which I'm currently now looking around to see if I can find a ruler. And of course, because I need one, I don't actually have one to hand. So don't be like me. If you are actually going to start a paper, make sure you have all your equipment. It's a funny one because there's so many students who actually turn up to exams and don't have the right equipment for the exam. So do make sure you start with the right equipment, a pencil, a sharpener, a ruler, a calculator, and obviously a pen. Now, I don't have the ruler. I mean, I could potentially use a ruler that my daughter has actually made because we always move and lose things, but I'm not going to use one. So I'm just going to assume I have used a ruler and look at what it measures as being. So the important thing for you to be able to do is to identify the size of the scale bar. So you'd use a ruler. So let's just say, for argument's sake, I'm going to say that that ruler is measuring, if I were to put my ruler here, it was measuring to 2 centimeters. So if it measured to 2 centimeters, what do I need to do then to do the calculation? So my image should go at the top of my triangle, my magnification. So I'm trying to work out the image length in the micrometers by using the scale bar. So therefore, I know my magnification because it told me it was times 1,250. So I know that this is times 1250. So that's the first thing to note. I already know that. Then what I need to work out is, right, okay, well, what does that mean when I look at my scale bar? So I've made the assumption, because I've just made it up, that my scale bar measured 2 centimeters. So if my scale bar measured 2 centimeters, the first thing I want to do is I want to actually turn that into micrometers, because that is what has been asked in the question. The question has said specifically that I want it to be in micrometers. So I want to ensure that I convert from centimeters to micrometers. So if I'm going centimeters to micrometers, I'm going to times by 10,000. If I were going by millimeters to micrometers, I would times by 1,000. So make sure you know how to do that conversion, because that is important. That will be something that they ask you to do. So if I've got my times, I'm going across, so I'm going to times that by 10,000. Therefore, I've now got 20,000 micrometers. So if I know that my scale bar is 20,000 micrometers, and I know that I have got times 1,250, I then need to divide that together. So I am just going to double check. And that would give me, so I would do my 20,000 divided by my 1,250, and that would give me an answer which is 16. So by using that measurement, that scale bar, I was able to work out that the length would be 16 micrometers. Now that's not the actual length in the mark scheme, because I have got it as a different length because I've either printed it out differently, or I have got it on my screen and I've used a ruler on my screen to measure that. So be aware that it might not match up with the mark scheme when you are trying to do this yourself. So that's one of the things that I would say is important for you to note. And as a teacher, when I set mock papers, I adjust the size of certain diagrams because I do that to check whether my students have actually memorized the mark schemes or not. I also switch up what I'm going to ask. So be aware, your teacher may do that as well. The correct answer they were looking for in this question was actually 12, but they will actually allow a little bit of deviation because it's about you measuring using your ruler. But the other thing to note as well is that they will give marking points for doing the calculations correct. So even if you get the wrong measurement like I did, I put it was 2 centimeters, I therefore would have lost that marking point, but actually because I did the right calculation, I would still get the marking point. So be aware if it's worth, for example, 2 or 3 marks, show you're working because that will allow you to get a mark. Okay, as long as you've done your calculations correctly, that will carry on. Oh, and look at this question. The next question was about labeling. B, which I'd already done when I looked here, I'd already labeled what B was. B is an erythrocyte. Now, it depends in the exam. Sometimes they will accept site you to put red blood cell, but some years they don't. So do be careful. Try to use the correct terminology when you are answering these questions because it will make a real difference to you getting the mark or not. Okay, so now they want us to look at the next bit. Cells A, C, and D, oh look, work together to carry out an important function. Now, I already said the fact I can see that they're cells, and also this one they haven't labeled up here. Let's call this one E. I can tell that these are my white blood cells, and they're therefore involved in that fighting of the infection. And I can tell that because this one that they don't want us to look at is a neutrophil, because it's one that you should be able to identify, and you should be able to identify that neutrophil because of the lobed nucleus. So it has a lobed nucleus, and I can see that because of this purple dark stain. We've got a dark stain here. And therefore, I know it's a neutrophil because of that lobed nucleus, which has been stained using a specific type of stain. So let's just go back to the question. What's the function? So what is the function? You'd be surprised how many students actually write the wrong answers here because they don't read the question. So they're asking, what's the function of A, C, and D? And again, we can also see the lobed nucleus here. It's not as clear, which is really interesting. This one's not as clear that it's connected, but it is. There's a very tiny connection just there. So it is important to note that one as well. Okay, so what's the function of this? Well, the function is it's going to result in an immune response. So we're going to have an immune response that is going to be working to fight against any pathogens that may come in. So our neutrophils are parts of our phagocytes. They're a type of phagocyte, which is going to fight an infection. Okay, let's have a look at this question then. So the smear has been stained with hematoxin and eosine. So this is talking about stains. So let's just have a look at our section on stains here. So we use stains to increase contrast and to make these structures more visible. And there are different types of stains that we need to use depending on what it is that we want to actually visualize. So as it says here, different cellular components have distinct chemical properties, the charges of them, the polarity of them, the solubility of them, and therefore different stains will be able to move. And if they can bind to whatever that structure is, they will bind to it and therefore make it visible. So you're told in this question that the smear has been stained with 2 stains. So we have hematoxylin and eosine stain. Now you're then told that hematoxylin stains nucleic acids blue So that's an important hint they've told us. And they say that eosine stains are proteins, and this could be cytoplasmic proteins, pink or red. So they're giving you little hints in the question. So it's important that you use that to help you. And let's look at what the actual question says. So explain why it's important to use differential staining. Now, remember, as I said before, that differential staining is about increasing the contrast. So in an exam question like this, specifically, they want me to say to increase contrast. And you'll find that that will be underlined in the mark scheme because that is a word that I must use when referring to these processes of using differential staining, because I want to see things differently. I want to see this one against that one. And so therefore, if I look back at this picture, I can quite clearly see I can distinguish A from C, from D, from B, because I have used differential staining. Now, the differential staining, as we said before, the eosine is going to give our cytoplasmic proteins a pink or red color. And if we're looking at our nucleic acids, the hematoxylin is going to give them a blue-purple color. So because of that, we are able to see where there is a nucleic acid being held inside, as we know, our nucleus. Now we can quite clearly see our erythrocytes don't have any dark blue or purple. So that allows us to see the erythrocytes without actually them containing a nucleus, because they don't, they've got rid of it during the differentiation process. And so we can actually see them without any stains. Whereas when we're looking at the leukocytes, when we're looking at our neutrophils, our macrophages, our lymphocytes, what we're actually noting here is that these white blood cells have got their nucleic acid contained within nucleus. And we can see that because we can actually identify them, A, C, and D, compared to the B. So it allows us to be able to count. So we can increase the contrast. We can actually see organelles, i.e. the nucleus, and that allows us to be able to identify these structures, but also we are able to distinguish different cells. Cells. And so it's a 3 marker that they're looking for here. And what am I actually distinguishing? Well, I'm distinguishing my leukocytes. And what would I do? I would count them. So if I want to, for example, have a blood sample taken, that blood sample that's going to be taken would be sent off. They would do a blood smear and they would actually count the number of blood cells that are visible in the blood smear as a way of indicating if I have, for example, an infection. Because if my leukocyte count is greater than what is classed as whatever the norm is, that therefore indicates some form of infection. So that could be something that is used as an identification of a patient having some form of problem. Okay, let's carry on because this question continues on. So again, this one, we're still using our insert. So we need to go back and look at the insert again. So make sure you're always going back. So not every exam will there be an insert, but for some there will. So you do need to make sure you're going back and looking at it. And the information provided, and that's the information above. This is the other thing I find really bizarre, that some students don't remember to look back up at the previous question. These are all sections within question 2. So with OCRA, they give you short questions, but the question goes all the way back to whatever that starting number is. So always go back up and double check. There's no hints in the question to help you. So the smear obviously was stained with the hematoxylin and it was stained with the eosine. So what do they actually want us to do with that information provided? So suggest and explain why the cytoplasm of C and D reacted differently to the stain. So the cytoplasm, well, we know we're not talking about the nucleus. So this is important. We know we're not talking about the nucleus here because they've asked specifically about the cytoplasm. So if we're looking at the difference between the two, we're looking at the cytoplasm. So we're not looking at this section here, this section here, this section here. We're not looking at that because we know that that is the nucleus. Now, going back to the question, it specifically asks about cells C and D. So therefore, I need to make sure that I'm not talking about A. So I'm only talking about C and D. And what's the difference? Well, the difference is this one actually does have a dark color to it, which is a purpley color. And this one has got a red color to it. So therefore, I can see, going back to the information that they told me, reminding myself, hematoxylin stains nucleic acids blue. So this one, we have nucleic acids. Nucleic acids, which are in the cytoplasm. And this again is where a lot of students get confused. They're like, well, nucleic acids are only held inside the nucleus, aren't they? No. You have to remember that through the process of transcription, the DNA gets copied onto mRNA, and that mRNA is made up of the nucleotides, which make up our polynucleotide. That mRNA strand, that polynucleotide, is a form of nucleic acid. So that is something to remember, that there is nucleic acids in the cytoplasm because mRNA obviously has to leave the nucleus to get to the cytoplasm. Also, you've got to remember that tRNA, when we're doing translation, the tRNA is also made up of nucleotides. It's a polynucleotide. It is a nucleic acid. So don't get hung up on the fact that DNA is the only nucleic acid within the cell, because it's not. DNA is the nucleic acid which is held inside the nucleus. mRNA can leave through the nuclear pores. tRNA is located in the cytoplasm and can therefore be found in the cytoplasm too. So that is important for you to note. And if we're going back to the question then, suggest and explain why cytoplasms of cells C and D reacted differently. They react differently because they have different structures in both and different concentrations of those structures. So we therefore want to use the term that there is more of our nucleic acid in C because that is a dark color, that is a blue-purple color. And those nucleic acids, as I just said, we're talking about the likes of our mRNA. We're talking about potentially our tRNA. Oh, and there is another one. The ribosomes that are found in the cytoplasm are made up of ribosomal RNA, rRNA. So therefore, we can say that cell C has got more nucleic acids, tRNA, mRNA, rRNA, and that is why it is a blue/purple color. So don't get confused. This is the thing that most students do. They will actually start talking about the nucleus because you will lose marking points when talking about nucleus because it specifically said in the question, the cytoplasm. So it is really important that you are taking the time to read the questions that are being asked of you. Then we need to look at, right, well, now we've talked about C because it is a 4 marker. We need to make sure we're talking about cell D as well. So I've done C, I now need to do D. So if I look at cell D, D is red in color. Well, what does that indicate? Well, we know it said above that, if I just go back up, refresh my memory, pink or red is to do with proteins. So I can say that D has more proteins in the cytoplasm. And what proteins might they be? Well, we're talking about cells that are to do with fighting infections. So therefore, they could be enzymes. If we're talking about our phagocytes which are going to produce our enzymes, which are going to be used to break down the pathogens. So the enzymes could be lysozymes that have been produced, or if, for example, we are looking at not a phagocyte, we might be looking at a lymphocyte, and it could be a T lymphocyte. If it's a T lymphocyte, it might actually be producing perforin if it is a killer cell. So it's important that when you are describing why, or suggesting and then explaining why the cytoplasm has reacted differently, you're using the information. It's about the proteins that are present because it was red or pink in color. What type of proteins could be being produced? Well, what if it's a B cell? If it's a B cell, it could actually be producing antibodies. So make sure that you are not only looking at the information that's been given to you, but also looking at what you already know and how you can then apply that to the question. So I hope that's been helpful for those of you who watch this video. And please do, if you have any questions, put a comment below, like it, and also share it. The more people who get to see this, the more people I can help. And I'll try and do more of these as we continue. Thanks for joining me.